You start then? It's all yours now. Okay, assalamu alaikum everybody. I know it is little late today, so and everyone may be feeling hungry also, so I will try to be short in my lecture, but not in content. Just I'll go a little faster, and you will have the slide. आपके पास सारी slides होंगी आप जो है वो तो स्लाइड्स जो है वो आपको मिल जाएंगी कोई भी क्वेश्चन हो अभी मैं हूँ यहाँ आई थिंक वी विल बी हैप्पी टू आंसर ऑल दोस क्वेश्चंस तो आज का टॉपिक जो है वो है टारगेट आइडेंटिफिकेशन वैलिडेशन टारगेट आइडेंटिफिकेशन पूरा आ रहा है यस वैलिडेशन ऑप्टिमाइजेशन ऑफ क्लिनिकल कैंडिडेट लेट्स गो डायरेक्टली टू दी एट्रीब्यूट ऑफ टारगेट तो कल हम लोगों ने बहुत बात की कि टारगेट है क्या हर आदमी ने ऑलमोस्ट जिसने भी लेक्चर दिया उसने कहा कि टारगेट क्या होता है तो आप लोगों ने आंसर भी दिया प्रोटीन है टारगेट विच इज़ ट्रू प्रोटीन इज़ द टारगेट whether it is G protein coupled receptors, whether it is enzyme, whether it is integrin, whether it is ion channel, these all are in our body, on the cells, inside the cells, outside the cell, on the tissue, these are the targets. So target identification is the foundation of drug discovery. Target has confirmed role in pathology and everybody can see that, right? This is a smaller screen, so you can see that. So target expression is not evenly distributed throughout the body. So brain में कोई target है, heart में कोई target है, but they are protein. I will come to that little later. तो टारगेट जो है 3D स्ट्रक्चर्स यूजुअली होते हैं प्रोटीन जो है वो 3D फॉर्म में होते हैं चाहे वो आयन चैनल हो चाहे GPCR हो कुछ पिक फोटोज़ आपको मैं दिखाऊंगा तो आपके समझ में आ जाएगा so target is assayable it should be there are targets which cannot be assayed we call it undruggable target तो वो भी हम बात करेंगे थोड़ी बहुत उस पर कि कैसे उसको जो है बाद में हमारे जब मैं आऊँगा अपने दूसरे लेक्चर में तो Dr. Varsi okay they should be relevant and they are very Dr. Varsi okay they should be relevant and they are very relevant to pharma industry we have one target I think in next last lecture today please stick around you will really see lecture today please stick around you will really see the real scope of the target how same target so artificial intelligence एंड फार्मा इंडस्ट्री को कैसे अट्रैक्ट किया उसमें ये मैं लास्ट लेक्चर में आज बताऊंगा सो आर्टिफिशियल इंटेलिजेंस कल भी बात हुई आज मैं भी बात कर रहा हूँ तो आर्टिफिशियल इंटेलिजेंस का क्या रोल है आजकल हर आदमी आर्टिफिशियल इंटेलिजेंस के बारे में बात करता है तो देखें आर्टिफिशियल इंटेलिजेंस जो है वो इससे जो है आर्टिफिशियल इंटेलिजेंस पहले नहीं था इट वाज नॉट देयर आई थिंक इन लास्ट 2 to 3 years we started knowing about artificial intelligence but medicines was there before also so it means that without artificial intelligence also you can discover a medicine as simple as it is but why we use artificial intelligence now because we can speed up the process we can reduce the cost of the developing drug and when cost of the developing drug is reduced it means when you take the medicine you get the less you have to pay the less amount so that's why we use artificial intelligence अब उसमें यह है कि ज़रूरी नहीं है कि हर चीज़ में हम आर्टिफिशियल इंटेलिजेंस यूज़ ही करते रहें कहीं पर नहीं भी यूज़ करेंगे तो अगर आप बीच वाला जो है मैं ये देखें तो आर्टिफिशियल इंटेलिजेंस मशीन लर्निंग एंड डेवलपमेंटल लर्निंग है अब आर्टिफिशियल इंटेलिजेंस यूज़ होता है सर ने डॉक्टर अजीज़ ने कल आपको ये सारे टर्म्स बताए थे और इनको एक्सप्लेन भी किया था एपिजेनेटिक प्रोटियोमिक जीनोमिक्स मेटाबोलॉमिक्स तो ये सब जो है वो आर्टिफिशियल इंटेलिजेंस के ज़रिए भी यूज़ सकता है एंड विदाउट इट आल्सो वी कैन यूज़ दैट तो ऊपर वाली अगर लाइन आप देखें अगर आपको दिखाई दे रही दे दी हैं ऑलरेडी सो डिजीज़ एसोसिएशन प्रिडिक्शन टारगेट प्रायोरिटाइजेशन प्रोटीन स्ट्रक्चर डिटर्मिनेशन एंड ड्रगेबिलिटी प्रिडिक्शन तो अब ये जो है एक जो हमने कल दिखाया था आपको एक ग्राफ वो डिफरेंट था ये डिफरेंट ग्राफ है ये आर्टिफिशियल इंटेलिजेंस कैसे यूज़ हो सकता है ड्रग डिस्कवरी में ये यहाँ पर ये दिखा रहा है आपको अच्छा उसके बाद ये जो है उनके एट्रीब्यूट्स हैं अब उसमें आर्टिफिशियल इंटेलिजेंस हम ड्रग स्ट्रक्चर बेस्ड ड्रग डिज़ाइन बिना आर्टिफिशियल इंटेलिजेंस के भी यूज़ कर सकते हैं ज़रूरी नहीं है कि आप आर्टिफिशियल इंटेलिजेंस में ही जाएँ उसके उसके बिना भी यूज़ कर सकते हैं। हमने अपनी पूरी लाइफ जो है वो कंप्यूटेशनल मॉडलिंग यूज़ की, कम से कम 30-32 पेपर पब्लिश किए। हमने आर्टिफिशियल इंटेलिजेंस वहाँ नहीं यूज़ किया, लेकिन अब हम यूज़ कर रहे हैं। अभी एक पेपर जेबीसी में पब्लिश किया, उसमें जो है और उसको पैटेंट भी कर दिया है, तो हमने आर्टिफिशियल इंटेलिजेंस भी यूज़ किया है उसमें। अब ये अच्छा है भाई। हाँ, मैं थोड़ा मूव करता हूँ, खड़े रहना जो है बड़ा मुश्किल है। हमारी आदत है कि हम स्टेज से बाहर आकर आपके पास भी आ जाएंगे, ये बहुत अच्छा है। ये आपने अच्छा किया। अब मुझे अपनी परेशानी नहीं होगी, अब मैं पानी हाथ में ले लेता हूँ ताकि मूव करता रहूँ। तो अब देखें, स्ट्रक्चर बेस्ड ड्रग डिज़ाइन है बिल्कुल लेफ्ट में आपके। उसके बाद आता है मॉलिक्यूलर डॉकिंग एंड सीक्वेंस ड्रग डिज़ाइन। तो मॉलिक्यूलर डॉकिंग के बहुत सारे टूल्स, अजहर साहब ने कल, डॉक्टर अजहर ने कल एक पूरी लिस्ट बताई थी। उसमें हम लोग जो है, क्योंकि इंडस्ट्री में काम किए हैं, हम लोग $1 मिलियन पे करके लाइसेंस करते हैं। आपको ज़रूरत नहीं है। 5 मोल, स्विस मोल, श्रोडिंगर का भी जो है पार्ट आपको मिल जाएगा थोड़ा बहुत। आप लोग उसको जो है डाउनलोड करें, प्रैक्टिस करें उस पर, कुछ भी करें उसको यूज़ करें, देखें कैसे, दैट्स इम्पॉर्टेंट। यू हैव टू लर्न योरसेल्फ। मैंने कभी कोई भी क्लास नहीं लिया आज तक, अभी तक मैं खुद ही करता हूँ जो भी करना होता है। और जब यहाँ फंस जाता हूँ तो कुछ दोस्त हैं अपने, और हमारे यहाँ कंप्यूटेशनल मॉडलर हैं, वो जो है वो आगे आ जाते हैं, वो सॉल्व कर देते हैं प्रॉब्लम। सो दिस इज़ आल्सो इम्पॉर्टेंट। और उसके बाद फार्माकोफोर मैपिंग। अब फार्माकोफोर आप लोगों ने अभी, आज आप कौन सब लोग गए थे फार्माकोलॉजी डिपार्टमेंट, तो वहाँ कुछ फार्माकोफोर के बारे में बताया आप लोगों को थोड़ा बहुत? नहीं, कोई बात नहीं है, तो इतना टाइम कम था इसलिए नहीं बताया। तो फार्माकोफोर इज़ ए पार्ट ऑफ ए मॉलिक्यूल, जैसे नाइट्रोजन, हाइड्रोजन, सल्फर ये है। ये कहीं पर आपके प्रोटीन में एडजस्ट करता है, अपने को अटैच करता है, तो हम इसको फार्माकोफोर मॉडलिंग कहते हैं। थोड़ा सा पढ़ना पड़ेगा इसके ऊपर। तो ये जो है वो बहुत इंपॉर्टेंट है, इस पर पूरी ड्रग डेवलप की जा सकती है, और कुछ भी ना यूज़ करें, सिर्फ फार्माकोफोर मॉडलिंग करें, यू कैन डेवलप ए ड्रग वेरी ईजिली। उसके बाद जो है डी नोवो ड्रग डिज़ाइन, ये मेरा एरिया है। मैं पूरा का पूरा अभी तक जितना मैंने काम किया डी नोवो ड्रग डिज़ाइन की है। खुद डिज़ाइन किया है मॉलिक्यूल को, हम प्रोटीन जानते हैं कि कैसी है। और मैं कल बता रहा था कि एक दिन जो है मुझे एकदम से हुआ कि मैं, ये प्रोटीन का स्ट्रक्चर मुझे पता है, एंड आई डिज़ाइन ए मॉलिक्यूल वर्चुअली, एंड यू कैन डू दैट व्हेन यू गेट लिटिल बिट ऑफ एक्सपीरियंस, यू कैन डू दैट। और फिर हमने उसको जो है, क्योंकि देखा कि हमारे पास क्या रिसोर्सेज हैं, उसको सिंथेसाइज किया, एंड इट वाज वेरी एक्यूरेट, एंड वी पब्लिश दैट पेपर। सो दिस इज अनदर पार्ट ऑफ दैट। ड्रग टॉक्सिसिटी, ये भी एक इम्पॉर्टेंट है कि हर ड्रग जो है वो पहली ही बार ड्रग नहीं हो जाता है, इट डज नॉट बिकम द ड्रग। ओके, सो आर्टिफिशियल इंटेलिजेंस इन दिस केस विल हेल्प यू अ लॉट, drug toxicity especially, because they can filter out what is the problem with this molecule. तो ये है। और उसके बाद ड्रग टारगेट इन, फार्माकोफोर मैपिंग इज ए computer uses pharmacophore to dock. It is computational modeling can be very different than pharmacophore. You don't have to think about this message in terms of filter. In pharmacophore you have to think about it. You can hear that, right? I have this. थोड़ा ऊपर लगा तो नहीं, मेरा है कि मैं मूव करता हूँ। आप घबराएं नहीं अगर मैं आपके पास आ रहा हूँ, आई जस्ट ट्राइंग टू कांटेक्ट विद, आई कांटेक्ट करना चाह रहा हूँ। उसके बाद टारगेट आइडेंटिफिकेशन, हमने कल बहुत बात की है इसके ऊपर, तो मैं इसमें ज़्यादा डिटेल में नहीं जाता। अब अभी मैंने थोड़ी देर पहले आई टॉक टू यू अबाउट प्रोटीन। तो प्रोटीन आती कहाँ से हैं, और कितनी प्रोटीन है हमारे पास, ये भी जानना चाहिए। सारी प्रोटीन जो है वो ड्रग नहीं हो सकती हैं, मतलब ड्रग टारगेट नहीं हो सकती हैं। So just give you an idea, okay, from human to rat, there are more than 10,000 genes, and it is in human it is 23,000 genes exactly. Okay, then these genes can produce proteins. It can be, like we said yesterday, also today I'm saying, it can be enzyme, it can be GPCR, it can be integral part, or any other proteins, it can be anything. अच्छा, ये प्रोटीन जो है वो सेल के अंदर और सेल के बाहर दोनों बन सकती हैं। जो सेल इज़ देयर, सो दिस सेल हैज़ द ऑर्गेनली So these are the all targets we use where protein will come from. For example, Golgi apparatus, mitochondria, ribosome, endoplasmic reticulum, nucleus. You know, DNA is a target for nucleus. So you can have the nucleus there. Okay. So we know about these proteins. So for example, let's have 500 proteins. These days, only out of 20,000, out of 4,000 proteins which are known to cause medicine, only 400 are targeted. So there are only 10% protein are targeted these days. So you have, so I did not put the number, but I just say that, okay, 500 proteins are being targeted. So you have like another 90% of protein is still not targeted, but they are causing disease, one of the disease. So now it is your job to find a nice molecule to target it. Okay, so this is, and then after that you go to profiling of the protein. You know, you have to do the assays, and you can develop assays. You can develop hundreds of assays, and then you can have the Dr. D yesterday said that 84 plates or 384 plates, or even now there are more than 1,000 miniature plates we call it. There are machines are there. They can use that where you have to use very nanogram amount of protein, and still you can get the result. Okay. So techniques. Now, I already talked about proteins. What are the techniques? How you can find which is the target identity? How you identify the target? Lot of things yesterday we talked. So bioinformatic approach, we talked about it. That bioinformatic is important. Also, you can use that, and like churning out the data. And we have good friends who can do that. Computational approach. Just now you asked about computational approach. Functional genomic approach. This is a new in Saudi Arabia, especially, and in US also. There is a lot of diseases which are rare diseases. In very few people are there. So we use the genomic approach, and Sir Dr. Azhar Aziz has a lot of paper on the new genes. He has discovered himself almost 10, 15 genes. So you can talk to him about that also. So that is a approach, also genomic approach. Artificial intelligence and machine learning, which I just talked about. High throughput screening, we also yesterday we talked. So we have a protein, then we have to develop or assay a large number of compounds. So how you do that? You have to develop a high throughput screening. I have some slides for that in the next. Protein microarray is also these days it is used for target identification. These proteins are very small, so they are in array form. Array form means in a plate form, and or on chips also sometime, and you can use that. Translational method approach. So from one gene is good for another discovery. So another disease. So that can be used. This is translational. In my third lecture, I'll talk about it. Phenotypic approach I already talked about, and in silico reverse docking. So this is one of the docking. So we just talked about docking also. Okay, target validation. So now this target identification. Now you got the target. Now how we'll know that it is a real target? So we have to validate that. It is like going to court and giving a witness, but judge will ask, are you a credible witness? So now we are going to do that here, that we have a credible target, and we have to validate that. So this is the similar. So these are the techniques. So I have to rush because I cannot. This every point requires one lecture, and Dr. knows that. Dr. Aziz knows that. So I'm trying to give you a flavor that how many things you can use, and just pick up one and master on one. You will be a very good drug discovery scientist. So that's what my advice to you. So target validation is critical step in drug discovery. It has suitable role, disease pathogenesis and therapeutic intervention. So genetic validation is one of them. That includes knock-in and knockout. I don't know in pharmacology did they talk about knockout and knock-in? No. Okay. I'll just use brief and then later we can do that. So that include knockout studies and knockdown studies. So we have to knock out one protein in animal. And animal is usually mice or rats. And we don't do in higher animals. So knockdown is also POLE, the expression of the gene then okay. So pharmacological validation, we call it inhibitors, activators, modulators. These are the ones which can be used for target validation. Animal model validation, knockout, knockdown, xenograph model, transgenic model. Anybody knows about xenograph and transgenic model? No? So xenograph model is we have an immunocompromised animal. So what we do, we knock down the immune system of the animal, and then we grow a animal. We grow a tumor of our trials, and that is called the xenograph model, right? And then transgenic model, we change the makeup of the animal. Dr.
Yeah, absolutely.
That, Dr. Zain is expert in that. He uses a lot of animals. Actually, he kills a lot of animals, right?
Yeah, we are a murderer.
So, so these are the target validation things, and you can use that. So now it goes to because the as title is going, I'm just going through title. Okay, so lead generation. So now we are coming from the target. So now we got the target, we validated the target. Now why? What we do with that? Any idea? Anybody? We have the target. We validated the target. But what do we do with that? So now we go to lead generation. So to target the molecule, we have to target the not lead generation, actually the target. Now we need something to work on that. So these are the outlines. I will give you a chart very quickly. What is the lead? What make a good lead? Screening approaches, of course, hit generation strategy and optimization. So these are all related to chemistry or biology. Or chemical molecules or biological molecules. So this is the chart where we say that lead criteria guidelines. This is the industry originated, so I will just walk through you one by one. So lead key requirement for the lead is it has a structural novelty. You need to have a novel molecule, and you can get a novel molecule by doing high throughput screening I mentioned before. Okay. Chemical tractability means you got a molecule. It should not be in air. I think it should be at least we can make it somehow. So that is we call it tractability. Okay. And it should be at least a real molecule, not just the phony molecule. Stability of the molecule, that is another. So these are all criteria, whatever bullet points are there, parameters. It should obey Lipinski rule. I met Dr. Lipinski. He was one of the scientists with Pfizer, and I put a question. Actually, now Lipinski rule is old. I will call it. We don't follow, right? We call it the Lipinski rule or beyond Lipinski rule. So I met Dr. Lipinski. He published this paper in Nature one time, and I said that due to you, a lot of drugs are not developed. And he said, read my paper. I wrote there. It is not a rule. It is a guideline.
Yes.
You follow guideline or you don't follow guideline. And that was his answer, and I read back again paper because Lipinski rule in our institutions they used to say you have to follow. And there are 5 points. I think I did not mention it here. No, I mentioned some of them. So the molecule should be less than 500 molecular weight. C log P is a parameter. It should be less than 5. Hydrogen bond donor and acceptor, it should be less than 10 and less than 5. So this rule we don't follow anymore. But there are, in some cases, especially in central nervous system, when you are developing a drug for hypertension or something, you have to follow that. Okay, and then for oral route administration, yesterday we talked about the route of administration, whether it is IV, oral, or nasal. This is parameters. It should have adequate affinity and efficacy, selectivity and primary and broad and So this is what you have to we have to do during lead formation. So and the other is developability enablers. So when we have the molecule, we can go in silico predictors also. We can predict a lot of things through that permeability, whether this molecule will be permeable through cell wall or not. This is another criteria. Another criteria. Hello, hello. Hello. Uh, Permeability P450. This is a liver enzyme. Okay, there are several liver enzymes. Some of them are called drug killer, like CA4 and C2C19. We call it the drug killers in industry. So there are many, like about 15 or 16 CYP enzymes are there, and we take care of that also. We do some assay on that. Protein binding is also important because when you take the drug, either through IV route or oral, there is the albumin or plasma or protein are circulating in your body, so these drugs they bind to that. So we have to see that there are three drugs also in your body: blood-brain barrier, HERG, and PGP. These are the parameters we usually use. And all these parameters you develop, and then you have to agree with your management also. That and they should agree with that. So for these criteria, we have the guidelines. Now coming to which compound to make. Yeah, I talked about the compound, right? So you have to make some compound. So for me, these are the starting criteria that we are starting with there. Okay. So if I'm lucky, I will make only one compound, which I never was, or nobody else was. So it is potent, selective, efficacious, and deliberative. Potent means it is potent on the target. Selective means it is selective on that target only, not the other targets. Okay. It is not. It is selective on one target, and it is not selective. Then it is not a good compound. Efficacious and developable. Developable means can be made into tons. That's another criteria we check early on. So which compound to make? Again, I just said here that if you are lucky, you have to make very few compounds. If you are not lucky, we are going to make 10,000 compounds we discussed yesterday. Okay. Now go to next so screening approach. This is the third part of this talk. Okay. So what are the screening approach? These are the screening approach we use after we found a target, validated target. We found a molecule. Now we have to do the screening to go back again. What is going on? So high throughput screening. We talked about it. That we have to develop a screen assay system which is very large. We can assay large number of molecules. So this is called high throughput screening or high capacity screening. Both words are used. Uh, Continuous screening. What is difference between high capacity and continuous screening? Continuous screening means we can pick up the compound like cherry picking and then do similarly. So that is called continuous screening. You don't have to do all 200 million compound or 2 million compound. Chemists can very nicely pick up the molecule, and we are all good in that. So we reduce the work and just go for few compounds. So continuous screening. Cross screening also, we cross screen between different targets. One target is kinase, one is 7 TM receptor or GPCR or CCP. Many other things are there. Then we do focus screening also. Once we have a compound, then we go for our target and see what happens. Select compound, then you heard that high throughput screening, millions of compound. Now we came down to where? Only 100 molecules. We are getting closer now. We are getting closer to getting a drug. So these compounds selected by similarity, by 2D and 3D, by computers to know about the ligand and target knowledge. So these are comprehensive, all works together. So once we have that, then we have to develop a full program to convince our company or organization that we are going to do high throughput screening. So screen adaptation.
हेलो
Just show you that how it looks on computer
हेलो
when we do
ठीक है ना?
that. So
ओपन कीजिए.
first thing, develop assay protocol. It may be 200 plate, it may be 2 plates. That's better now. So it may be 200 plate. I just put a number. Don't go with that number. It may be only 2 plate even. 2 plate of 96 well plate. So 200 compound only. Okay, then we get the primary heat out of it. So we set up the criteria for primary heat, like it should be 10 micromolar activity. Okay, and then confirmation we do that. Maybe we get out of millions of compound, we go to 20,000 compound, maybe 20 compound only. Then pick up the 2,000 compound from there, and then we do the IC50. IC50 means, anybody can give me what is the meaning of IC50? You had been to College of Pharmacy. IC50 means inhibitory concentration at 50%. That's the word we use for in drug discovery. So first parameter what we do is IC50, okay? Or sometime we do only percentage inhibition because we have large number of molecule. So we do single point assay. IC50 is a curve where we do escalating dose and see whether what is the 50% inhibition. But sometimes we just do the percentage inhibition, only 1 or 2 volumes, and then we know that compound is active or not, like higher concentration, lower concentration, there is something there in between. So this is also can be done.
Can you just check it? Give me that. I'll show you IC now. I'm
Excellent.
working on it. I will display that. Okay. Can you give me keep it for your just to know what is the graph.
Let me see, maybe I have the graph.
Yeah, if it is there, it will be good. Because that will connect a lot of things coming forward also.
I think you can repeat it.
No, I think I have IC50 graph. Just yes, I think you can explain it here.
Yeah, it's there.
Yes. Where it is gone.
Backside. Yeah. So sir is talking more of that, if you see a graph there, where when you do a cell base on these things, and then you try to do IC50, IC90, how much a particular concentration kill or act on the target, okay? That's basically the basis of whole research which goes on. Something IC50, some is required IC30 only, and KRAS is now, only IC30 works in a different, otherwise it becomes a toxic.
Yeah, it is system-based approaches. So I have example for system-based approach. So now chemists use, they call it privilege structure. So privilege means it is known to only few people, right? So it is very normal in companies. We hide the idea from other people and we don't tell anybody. So this is privilege structure, or scaffold also, sometimes we use the word. But sometimes it is published and you can make it out that what we are talking about also. There is a term we use in industry, competitive intelligence. I just want to tell you why I am telling about that. So competitive intelligence is spying on the other companies. No, he's not looking at me. No, I'm not stealing. So competitive intelligence is very much related to privilege structure. I tell you a story that in one meeting, one of my friend in American Chemical Society meeting, he wrote, there were a couple of friends from Pfizer and there we were sitting, and he wrote something on the tissue napkin. And the other guy said, oh, can you give it to me? I'll throw it out. And he did not throw, he put it in his pocket. And they went back, and after a few months, we know that they patented it. So this is the privilege. He told the privilege structure just on the dinner table, and the guy took it. Another story is about Robert Woodward and E.J. Corey. they still, Robert Wood died, E.J. Corey still says that I gave him the idea to get the Nobel Prize in long time ago, 75, and E.J. Corey got in 85. So he was very unhappy because he was the one who gave, and he said, I wrote on a piece of paper and I gave it to him. So if you are going to work in industry, don't write anything on the tablecloth. Remember that. Okay, pharmacophore, I already talked about it. Receptor modeling, I am going to give example. Exploiting existing data, we do a lot. This is a strategy to find a hit, okay? So you have to go to literature, keep reading a lot, and you will find something which other people have not done that. I will talk in my third talk. And that data you can use. How you will use, you will learn slowly. Exploit new validation chemistry to increase diversity. This is for chemists, we always look for new way to make. Like, for example, this mic is this mic is like that, but someone else mic make it like that. So this is the existing mic doing the same job, looks different. So that's what we do here also. Okay, I said that I will talk about knowledge-based lead discovery. So a system of family of protein with common motif that forms 3D space, and that they are receptive to a small organic molecule. So the picture you see, this is a computer-generated picture of a protein, okay? It may not be looking when you look at the microscope, but this is computer-generated, and we know what they are. So we get the knowledge from this picture, and we discover some small molecule. There are programs, algorithm has been written how to use that. So ligand for one target is applicable to other targets also. Same ligand can be used for some other target. Small changes here and there, and that can be good for the other disease also. SAR, we already talked about it, and SAR-H provides mean to predict the active small molecule for other target system also. So same, very small changes in the protein or in the molecule, it will be good for other target. System-based research, I said that I will talk about it. So this is the system-based research. You see all the protein I already mentioned to you, and how we go there. So in this case, biology, bioinformatics, or informatics and chemistry, they work together to develop a system which is called SBR in the middle of the this graph. And the proteins are here: protease, nuclear receptor, 70M or GPCR, kinases, ion channels, integrin. Dr. Azhar, actually, he gave the list of these yesterday, all these proteins. So this is, we call it system-based research. And we use a lot in drug discovery and target validation, target finding. We use all these system a lot. Okay, knowledge-based hidden generation. So this is a complicated little bit, but you have to see the arrows going all over. It is all integrated, okay? And that's what we say, that in AMU or any other company or institution, we have to integrate with each other. Then we will be successful. You cannot work in silo in one place and just do yourself, okay? So this is I'm not going to explain that, but this is, you can look at that how this system works, okay? And knowledge-based hit generation process works like that, okay? And this develop a system knowledge and knowledge-driven design and synthesis of novel chemical targets. So we use some of them, or sometimes all of them. We do that. Okay, receptor modeling, I said that I will give one example of receptor. So this is our work, old work from GSK. So P38 is a kinase inhibitor. You can see the inhibitor is right here. So we have to change it. This was not very good, okay? So we have to change it. So we have to replace few things from here, replace trying to make hydrogen bond difference, and because it was not solubility was a problem with this. And it may be due to this molecule or this part or this, amide part, it was not stable also. So we used some of the tools which we have. novel isoster for biphenylamide. So isoster is a term we use to replace one of the ring or one of the functional group. This is a whole new science, a whole science. It's very easy to do that. So then we solve the X-ray structure also of the molecule. This is the X-ray structure molecule bound into protein. Now we know where are the contact point, and we can replace very easily something which we know that it is not stable. So now P38 enzyme, we made about 347 molecule, used array to test all those molecules. Then we know that the problems were there. So from this we made this molecule which was stable, and this became a drug molecule. And then we did the crystal structure of that molecule, and which is like the previous one. So now looking at this structure, and this is, you see the change. The change is huge here, right? But it has the similar, if you see the PIC50 or IC50, in England they call it PIC50, inverted log actually. So this is like 7.4, maybe about 9 nanomolar or something like that. It is same, but the compound was very, very stable. It is soluble, and solubility comes from nitrogen also. We will talk more about that. So we changed the molecule completely. We took out one imide bond, which is important. We don't like too many imide bonds because they can be hydrolyzed very easily. And we have a new ring system. So this is how we do that. Okay, heat generation summary. Just let me, whatever I said so far, I will just summarize and then go to next step, lead optimization. So efficient lead generation requires high throughput screening, computer-aided structured drug design and discovery, and need to make a lot of molecule. As a chemist, we always say that make more molecule. Every molecule is $1 million or $1 billion drug for us. So you remember that. We have privileged structure, we have pharmacophore analysis. I already talked about receptor modeling, I talked about it. Exploiting existing data system, that is also, I gave a brief idea about it. New chemistry, we always look for new chemistry. Yesterday, Dr. Aziz mentioned about Barry Sharpless. Barry Sharpless, the only person who got 2 Nobel Prize in chemistry. Linus Pauling also got 2 Nobel, but one was peace and one was chemistry. But Barry Sharpless, I met him in Singapore. I was sitting next to him, and I asked him that how much time you spend on computer, not these days, computer. He said, I spent 2.5 hours on SciFinder every day. And I said, how you find time, Dr. Sharpless? He had, at that time, he didn't get the next Nobel Prize because he had the 2 Nobel Prize. One was on hydroxylation, asymmetric hydroxylation, which changed the whole industry. And the next one was on click chemistry with Caroline Bartozzi. So he said, I spent 2.5 hours every day to read literature. I think you have to make habit of that. If you want to become a good scientist, good drug discovery, and you want to discover a good drug in this country, you have to read more and more. So this is the Nobel laureate, he told me personally. He spent 2.5 hours every day. I don't spend even that much, so I didn't get the Nobel Prize, that's why, right? Okay, so, and sometimes we purchase the molecule also. So why we purchase, why we don't make it? Because molecules are sometimes, there are consortiums and they are selling molecule to us, and we pick and choose and we buy molecule, we make a big library. So this is the heat generation summary. Lead optimization, I think I'm going to just end here because I'm going to give lead optimization. Stay tuned in my 3rd lecture, which I'm going to talk about case study, and I'm going to discuss all these points you can see here. It will be really interesting. Stick around, okay? Stay here. This will be my last lecture today, and it will be a little longer also. So what I'm going to talk is what is the lead optimization, how we do that, screening cascade, I will talk about it, molecular interaction, I will show few things about this molecule which is already in phase 2 clinical trial, and we have started a company on that. modulating property, how we modulated the property, how we got the molecule. A whole process I will show you, inshallah. And then medicinal chemistry strategies, because I am medicinal chemist. If I don't show you molecule and synthesis, then I will not be very happy with that. So I will show you how, from where we started and how we went to the end. So this was my last slide, because the lunch time, it is getting late, so I took only 35 minutes today for this. So if any questions, I will be happy to answer.
I have one question.
Yes, sir. Please.
I think this drug discovery has multi-factor, multi-level kind of, where you have lot of factors and there are lot of levels affecting your drug design of the molecule.
True.
So, are you using any kind of statistical software or tools to reduce the number of experiments?
Yes, I already mentioned AI is helping us. There are some tools, especially in suits like Schrodinger or molecular operating environment, where we put our molecule and see what is the, for example, I can figure out what is the toxicity of this molecule. If it shows high toxicity or a number of molecule, maybe let's put 100 molecule I put there, okay? And then the computer will tell me that don't make out of 100 80 molecules, make only 20 molecules, which is doable. So there are tools already, and there are some of the tools are already available online free. So we can start with that. We use, because we had the license, so we use Schrodinger and molecular operating environment.
Design of experiment.
Design of experiment are usually, it is set, like assay, what you will do. In case of assay, we know how to do that assay. We cannot reduce the time there. We can reduce the number of assay, but assay is set. Yeah. So to my knowledge, we cannot reduce the assay. Nowadays, what AI is doing, AI going a little bit further, okay? They say that we are going from, they don't have to do hit generation or hit or target, but they will give you a target. They will give you a hit also. And they say that this is going to be phase 1 molecule. Now, we do make the molecule, but we cannot go, the regulatory agency is still, they want animal model done. It is that time has not been reduced. Only 2 days ago, somebody posted that AI has reduced the drug discovery time from 12 years to 20 months. And there were like 100 emails, 100 hits there that you are lying, because you can reduce the time from where you get a target and get a molecule. That time can be reduced. But when you dose the animal, you have to do for 5 days, 10 days, 2 days. You can reduce that time. If you get in animal, you cannot reduce the time of safety in human, which is phase 1. I talked about this. You have to do like 48 hours to 7 days. And we are setting up a phase 1 trial center. Actually, Dr. Azhar's office I broke down. I showed him the picture. He used to be, it was his office. We are breaking it and we are making 30-bed suite there. So that part you cannot reduce it. Still, computer is not that smart to do that. Maybe in future, I don't know, it may happen. But right now, we cannot do that. So.
Thank you. I think if you have any more questions, we can do it during the lunch.
During the lunch.
But we'll have to reduce the time now. Let's get back in 30 minutes. Okay, let's try to be quick.
Come and interact with us. That's not a problem. Okay.
We have an introduction session as well. We have not done the sampling. That what is your background? What is this? I think let's do this in this session.
Yes.
Before we go straight into the slides.
I like to know that.
Yeah. This will be
so that I can be more receptive. If I'm talking too high, then I can do it will be better.
Okay. So let's proceed for the lunch. It's ready, and we reconvene in 30 minutes. But don't compromise on your calories. Okay,
Don't ओके वो
नहीं नहीं,
हाँ
हम अगले अपने उसमें भी हमारे पास है, तो इसको अगर आप जल्दी रैप करेंगे
चलिए ठीक है हाँ
कैचअप कर लेंगे।
बिल्कुल ठीक है मैं जल्दी रैप अप करके और आ जाऊँगा अगले
वो वाला टॉपिक
वो टॉपिक इम्पोर्टेंट है वो ठीक है हाँ
थोड़ा ऑफ कर लो अभी के लिए, क्योंकि लाइव जा रहा है ना, तो आप
अच्छा
डिस्कशन करोगे वो भी
ओ
लाइव में जाएगा।
ओके ये
तो
I think inshallah 5 minutes okay
if you guys can check outside, if people are there, they can call them inside, if you don't mind.
So we can message on WhatsApp.
5 minutes
Okay,
we'll
yeah,
start
group you can message, yeah.
yeah your group you can message
Let me message.
Okay, Bismillahirrahmanirrahim. Assalamu alaikum, everybody. Let's start. I think it's time because we have one more lecture to go, which will be really interesting. The next topic is industrial and academic perspective of drug discovery. I will pass through quickly on this one because Dr. Zain also he will talk about it in his lecture by tomorrow, inshallah. So what is the outline of this talk is like that, but I'll try to give you a quick glimpse of that, and if you want, we can do it again if you want. The meaning of academic relationship, valley of death, significance of valley of death and industrial and academic perspective, how to overcome valley of death, example of industry and academic drug development, translational research and IP, and drive model, a successful academic drug model. So this will be the outline, but I will talk few things and then zip through some of them. So why it is so important, academic and industrial drug discovery, why it is important. So this is a renaissance is happening actually. You can call it a renovo in other Latin word. Industry and academic perspective, academia has, so what academia has and what industry has, that's important. So academia has state-of-art laboratories and technology. Academia has brilliant human capital, human mind. It does not mean that industry people they don't have mind, but the academic people have more chance to learn and they have more time without much more, they don't have that much pressure. Industry has brilliant human capital and deep pockets to invest, means they have a lot of money and they can invest in the drug discovery, which is, you have seen, it is very challenging thing. Industry benefit while institute learn about the business reality and market needs. What is the obstacle? And this valley of death is talked a lot these days in industry. Why? Because industry fails a lot to drug discovery. So who will help them? Academic people will help them. So this we call it a valley of death. The large gap between basic scientific research and transition to novel molecule need restructure education and academic research. Why you are here? You are from academic institution. Anybody answer me? You are from academic. Why you are here? You are filling the gap which we have, which is taking us down. So academic institutions are filling the gap where industry is failing, as simple as it is. Okay, I worked in academic and I worked in industry. I know what is the problem. So need to restructure education and academic research. So all academic researchers have to focus more towards getting a product. And the product can be sensor, can be drug, can be anything else. And cultivate fertile ground between academia and industry. So we have to work together. Industry has come and pay money to Dr. Azhar so that he can buy equipment, so that he can buy some chemicals, and then he can teach you the drug discovery. That's the part of the industry which they have to do. Then we will avoid the valley of death because industry fails a lot, so we have to fill the gap, and which is given in between education. That's the part. Okay. So drug discovery in academic faces specific challenges. I will just very quickly read it, and it will be addressed later by my colleague here. So lack of funding, lack of expertise in universities to assess potential. So like one professor was asking me just in lunch that what he can do. She don't have the resources which industry has. So you have to connect with some industry so that they can work on that. And a tendency to replicate the industry model in academia, that's a very bad thing to do. Please don't do that because the industry has a lot of money. You cannot replicate in an academic institution. You have to focus on your education plus idea and sell that idea to industry, okay? And lack of experience, academic institution, they don't have that much experience in clinical trials, which is very important once you have a drug. So That is another. What are benefits between collaboration? Scientific outcome, commercial outcome, and follow-up cooperation. So that's the benefit if we work all together. Now, examples, I will give you quickly some examples of industry academic collaboration. Examples of academic industry collaboration. This is like in between is the industry. One side is the academic institution. Other side is the non-profit organization. You cannot see it here. It is grayed out. So if all work together, then it is easy to climb. So there are three entities: pharmaceutical industry, academic institution, and non-profit organization. Who are, I don't know about India, but in US, Gates Foundation, Fox Foundation, in Saudi Arabia, Evolution Foundation. There's another one recently they're coming up. And there are people who have deep pockets. We call it angel investors. So they can also help. And I'm sure that in India also there may be some people are there. Pharmaceutical industry possess financial resources, funding large clinical trials. Clinical trials requires usually $500 million per drug. So academic institution like AMU or any other university where you come from, we are not ready to pay that. So who will pay? Industry will pay for that. Establish manufacturing and distribution network, which is not in academic institution. Commercialization strategies, infrastructure capable of treating patient population. On clinical trial, you need hospital. You need very, very specific, setup to do clinical trials. Our hospital in Jawaharlal Nehru Medical College, I'm sure that they are doing some, but that requires also a lot of investment. The average cost of drug development is more than $2 billion sometime. No academic institution can afford that. So we have to collaborate with the industry to get there. Okay, so pharma industry ensure that you can navigate regulatory landscape, ensure new therapy are safe and efficacious before reaching to market. So this is the job of pharma industry. You give them a molecule, you give them a protein, you give them an antibody, you give them anything else, a DNA, RNA, but it is their job to make sure that it is safe when it is reaching to patient population. I'll give you 3 or 4 examples and then I will end this talk. So there are examples of Novartis and Oxford University, development of gene therapy for spinal muscular atrophy. Novartis provided funding, expertise in clinical trial and regulatory affairs. Oxford team led the development of gene therapy technology. And what was the end result? There was a drug called Zolgensma in phase 3 clinical trial. And I think it is going to be approved next week or so. And projected is that per patient, they are going to charge $1 million. And there are a lot of them. So in US and other places, they are the insurance people are going to pay. I give you one example which I am involved right now. There is a drug gene therapy called Casgevy, which is made by Vertex, and it is being used in our hospital where next to our hospital, I'm in research center, there is a hospital there. This gene therapy costs $2.4 million per patient, dollars, in terms of dollars. And I was had honor to be involved in conditioning the patient, and this medicine was first time used in Saudi Arabia. First time used in the world outside clinical trial. Clinical trial is not the patient, they treat, but this is a trial. Outside the clinical trial, first time we used that, and I was involved in that. I think it's done. So I can speak. Yeah, it's done. So I was involved in that, and there was a news article and many things we were given. Accolades that we helped them. So similarly, in case of this Zolgensma, I'm sure that somebody has given the money to do that, and it is going to cost a lot. So this is the industry, pharmaceutical industry is going to pay for that. Academic institution, what they do? Often they are the originator of idea actually, most of the time. So they are the originator of idea and cutting edge technology. Uh, in front of, they are forefront of scientific. Hello. Yeah. So academic institutions are forefront of scientific discovery. Lot of discovery, I will say 80%, in my opinion, is done by academic institutions. Ideas and technology are uncertain. They discover, but when certainty that certainly it will go to market, it will be done by industry because they will spend a lot of money in that. But academic institutions have a strong ties with patient advocacy group. So what happens that if some new drug is approved, uh, they usually come to university hospitals. So those patients are used for the clinical trial. But industry don't have a hospital of their own. That's the problem. There is a university in Saudi Arabia called KAUST, King Abdullah University of Science and Technology. They always come to us because they don't have any hospital. This university is very famous. You can hear me if it is not working, right? Okay. This university is very famous. Now suddenly they get into medical research also, like us, but they don't have any hospital, so they are always asking us to do trials. बंद कर दे इसको। कम कर रहे हैं, कम कर रहे हैं। Okay, so example of messenger RNA vaccine developed by Kathleen Carrico, who won the Nobel Prize. I think most of us, lot of us, if not most of us, they have the vaccine. This was discovered in an academic institution. Moderna and Pfizer, they developed it. The most important part was the formulation, actually. So they did that. It was invented by academic like Dr. Carrico, who was one time at, there's a huge story about it. I think it is worth reading. She was not given any tenure position in the University of Pennsylvania. And she was the one who discovered, purely academic institution. First year, this vaccine was sold by Pfizer, 25 billion. Second year, 22 billion. They made huge amount of money. Now it is only 1 billion because nobody is taking that vaccine. So for 2 years, they made about $50 billion. Pfizer, Moderna also, they made a lot of money. I don't know how much. Marketed and manufactured by Pfizer and Moderna and Pfizer. Academic industry relationship, more examples like Center of Biome for Microbiome Innovation, Pfizer and University of California, San Diego, they have GLP-1 drugs for broader indications, applications, drug resistance. USCSDA provide microbial genomic information, and the discovery is done by the Pfizer. Collaboration to solve growing threat of drug resistance in the bacteria. Yesterday, Syed Ahmed talked about it. Mr. Syed Ahmed talked about it, drug resistance. Financially, implication of academic and industry collaboration. So traditional drug discovery time is long. It takes very long. Okay. Academic industry collaboration can significantly reduce this time. So as I told that academic institutions have a mind, they work harder, so they can reduce the time of drug discovery. Can be done by optimizing clinical trial design and do other things, leveraging existing partners. For example, partnership between Glaxo and Cambridge University. This is one example. Okay, neurodegenerative disorder, they have worked on that. Cost saving and developed translational drug on by Cambridge technology. There was a milestone was paid to Cambridge University, which was very huge amount. Accelerated approval of Vicara Park. This is a drug which was developed by these two institutions, and royalty payment to Scripps was really very high. So what they did, they took the money, invested in the research because it is an academic institution. So that's what between these two have to do. Return on investment. Industry academic partnership enhances financial return on drug development. Okay, they want, if they give you money, industry, they want something in return. It is not free. Or if you are as an academic institution give them, you also want return something. So that's the example between Amgen and Toronto. They developed Riptaka, PCSK9 inhibitor. University of Toronto used expertise in lipid metabolism, and royalty was in millions of dollars. And the University of Toronto developed a whole lipid institute by taking that money. Another example is Broad Institute, MIT, Harvard, and Bayer, which was my company. They collaborated, yielded 3 clinical candidates, and is still going on. So intellectual property, right and dispute, Dr. Rizvi is not here. He was asking a lot of things about. So for academic people, yesterday I said, again I am saying, please do not publish until unless you are sure that it is not of any value. Okay? Make sure that you are protected, your rights are protected. I worked in industry and I know I will take away your right, as simple as it is. So be careful. If you discover something, you talk to somebody like you are talking about your work, patent it. Or at least file a provisional patent, or at least you should write it down on a notebook, get a signature, your signature, and 2 other people should sign on that. That's evidence that you conceived it and you are going to develop it. That's very important. So that's the whole thing is right here. Collaboration, potential, present challenges. Challenges are related to institutional priority in both academic and industry. Academia and industry protect their both of them, they protect their invention. Nobody want to give anything else to other than and you should be paid. There are examples of UC Berkeley and Genentech who are recombinant human growth factor. This case was highlighting the defining of patent scope. Now, you are asking about composition of matter, and in this case, there was only one word was not written, and Berkeley got 20 million for that. Just one word was not written. So, and Genentech patented it. They said that you are infringing. हमारे patent पे तुमने कब्जा कर लिया है. They go to court, and court awarded 20 million to Berkeley, just for that. So it's very important that the patent should be written in a very broad way, as much as you can. Well, examiner will come and say, cut down this and cut down, that's okay. But you should broadly patent it. Pfizer and Washington University dispute is very well known. Celebrex is a drug everyone uses these days for COX inhibitor. So inventor came out and he said that this is my idea. So Pfizer said, no, it was not your idea. It was our idea, and he produced his notebook pages, and he was awarded large amount of money. Fortunately, one time I also had to go to US court to show that we were first inventor, but I did not get any money because I was working, and we sign a paper that you are working and anything comes to company will be for company. So we won the case also in that case. So my notebook was presented in the court. So notebook is very important. This is the time when we were written by hand. Now it is electronic lab notebook. So that is also accepted in court. Okay, this is my last slide, I think. A successful model is, this is Emory University. There's a professor called, his name is Dennis Liotta. He discovered most of the antiviral drug, okay? A lot of them, HIV drugs. He was the one who was inventor. And he was smart. He put this model. Okay, industry, you want money, you come and I'll give you my invention. Okay, so he had, like, I think I put the name of the inventions. That it was run by highly managed team and commercialized people. Okay, academic people, they don't know how to patent it, things, or how to commercialize it. So you have to, if you are starting something, please contact an attorney or somebody who knows about business and talk to them, and they will tell you how to go. It's very easy. It is not difficult. Don't go by textbook, okay? I know people are buying textbooks. You have to have a real life example from somebody who has done that. Like Mr. Sayyid Ahmed, who came yesterday. Did he see, you saw his first few slides. He went there, came down, then went up. So that happened a lot. So you have to be careful. This is example for me also that I learned from him. You know, we don't know how to negotiate with the company people. They have deep pocket attorneys and you even don't know how to talk to them. And can advance in-house drug the candidate to higher value? Okay. Sometimes we think, oh, we got it, okay? We are going to make money, but we don't know whether it is 1 million or whether it is 20 million. So you have to talk to somebody to get your real figure also. It is like buying a house. You go and the realtor will say it is 20 lakhs, but maybe it is 5 lakh only. So any invention, I'm not talking about drug discovery, I'm talking about stethoscope. Anything you think about it, you have to have a person who knows about it, or an attorney at least, they can tell you, okay? So this is the drive model which is being used all over industry now and academic institutions, and it is very successful because a lot of people are involved in that. With that, any questions? Yes, please.
Drive model that you have mentioned right now, is it specified for any university or is it completely
It is between university and academics. So this drive model was made by Dr. Dennis Liotta and his team because he discovered 3 molecules which were sold in billions. Cipla also made one of the molecules after patent expired, and they supplied to whole Africa. So he protected his right only. He's protected Emory University right. That's what his part.
so is it a private entity or it's
No,
something
it is not a private. It is part of the university.
exactly?
It is part of the. That's what I wrote. That Emory University. This is part. University is actually pharma company. I work in pharma company. He is working in pharma company. We are very bad people usually. Okay.
Yeah,
No, no, I'm just looking at. We are. I work there and I have done that. We are like vultures. Okay. I want to take away from you how you protect yourself. It is your job. So that's the drive model. Okay. So Dennis Yorota, he was smart. He said, he knew what happened to University of Berkeley. University of Berkeley would have got $1 billion. Okay, but they got only 20 million. Okay, but he was smart. So Emory University, they got $565 million for his inventions, and they set up whole new chemistry. So he is a hardcore chemistry person, so he set up a new chemistry building, very big building. So that's what you have to do. Okay, any more questions on this? So we can take 5-minute break. We can introduce each other.
yes, that's what
Okay, I took your word.
so, so is it possible that you can have a little introduction? It should be done earlier, but let's do it now.
Yeah, while we are taking 5 minutes.
Start from right. So next to you. Yeah, just tell me what department you are coming from and what is the purpose of doing this.
I am Nandini Aditya. I completed my master's in biology business from AMU. And during my master's project, my work project, I did work on biosimilars. So there my targets were drones, exceedingly long drones in connection to respiratory, and we found some specific molecular protein we made a library plant called in-house. So within it, we discovered some compounds called S inhibitor, CDK1. So I want to know more about it from
Good.
there. So
So
that's
you're
why
from
I
department biochemistry and you wanted to do some more research on what you did already on the research. Yes.
so sir, I have started. I am same from biochemistry department, and I have also completed my masters. Mm, during my master's thesis, actually I was my main project was
That's fine because see, this is not going to be very detailed, so that's fine. You are interested in doing that.
We just want to know what is.
Yes.
my main project was like
launch करने पे आकर इसको it is not launching. Oh, there is something. It is not showing up here. Whatever is there. Security alert. I think maybe another window is there. I think yeah, there is another window here. Small, I remember. Let me call. पीछे बैठे थे मुझे दोबारा लगा इसमें. Yes. It's working now. Okay. Okay, this is my fourth lecture of today. I believe this whole session. Okay, let's start on the last lecture of today, and this lecture is close to my heart because I worked on this project for a very long time, and I was very successful in this project. So I want to share the knowledge with you how we did. This work was done at Bayer in US, and when I was in Germany also for some time. So this is a compound which we developed, and I'm going to show you some chemistry also. So please, those who are chemists, they will enjoy. Non-chemists, you will also enjoy because I'll make it very simple for you how we develop this molecule. This molecule, which I'm going to, this is for CCR1 antagonist for anti-inflammatory and neurological disorders, and another one which I'm going to show you, and this is I'm going to show you first time. I have not shown it anywhere. What we are doing right now with this molecule, and you will see that how successful it was, and hopefully, inshallah, it will be more successful in the other indication. This molecule went 2 times phase 2 clinical trial, which is huge. It means every clinical trial cost us $200 million. But at that time, yesterday I mentioned it may fail, so it failed there. But we did not give up. We are not giving up on this molecule. We are still doing that. So CCR1, just give you overview, and I'll explain what is CCR1 quickly. Background of chemokine program. CCR1 is a chemokine. We talked about the surface proteins. This is one of the proteins, CCR1, chemokine receptor 1, we call it. Discovery of template through HTS, we talked about high throughput screening, so I'll show you a real example how we did that. This is all done by me and our group. Synthetic optimization, I was talking about optimization, how we have done that, you will see that. Then pharmacological data, performance in efficacy model, repurposing for other indication, we talked about it, and then summary of the result. So what is chemokine? Chemokine are cytokines. They are one of the proteins in the body, and they are involved in different biological processes or pathological processes. They have 70 to 120 amino acid, mostly basic proteins. So there's nothing special for that. There are 20 amino acid which are natural, and they encompass one of them or all of them. There are 2 major classes, CXC and CC. Why? Because CXC is lysine is there, so that's why we call it CC. Classified based on the position of 2 cysteine in the amino acid, and cysteine has a sulfur. That's important, so remember that. Produced by immune and non-immune cells, 45 chemokines and 20 chemokine receptors has been identified. There are more now, about 78 or so. This is old slide, so I did not update. But those are not that important. There are few of them are very important, and there are drugs for that in the market. They involve in biological processes like growth regulation, embryonic development, angiogenesis, HIV infections, and cancer also now. We just discovered about cancer recently. Chemokine and disease, they are involved in normal immune response, multiple sclerosis. We have done multiple sclerosis is a neurological disease. We have done a clinical trial on that. Organ transplantation, rheumatoid arthritis, atherosclerosis, and dermatitis, and cancer. Chemokines and multiple sclerosis, this disease is mediated by T and B cells. Those who are in biochemistry, they know what are the T cells and B cells. They are lymphocytes, macrophages, resulting in extensive inflammation and demyelation. Inflammation, you know that demyelation means on the nerve endings or on the nerves demyelation takes place, and then your nerves are exposed. So that is called demyelation. In 1995, there was a model was developed. I think somebody was talking about, that's what I asked, that dementia model. See, these models are not easy to develop. It takes months and years for someone to develop. So somebody developed for us this model, and MIP1 alpha. So every protein has an indigenous ligand also. So like chemokine, indigenous ligand for that is MIP1 alpha or macrophage inflammatory protein alpha. So we have to stop interaction of MIP1 alpha to chemokine, and then disease will not spread. Or a peptide or a protein. Okay, this is just nutshell. I'm talking about the mechanism part. Okay, so for us, we were given a goal that we had to come up a small molecule, not a peptide, to develop a antagonist for the CCR1, in the hope that we can get a new therapy agent for MS. Earlier it was MS, then it became endometriosis, then it became transplant rejection, now it is multiple myeloma. And we have done any models in every, clinical trials in every case. So I talked about high capacity screening or high throughput screening, both are the same word. The Berlex compound, Berlex was a Bayer company at that time. So we had a library, big amount of. So we screened it. We developed the assay and we screened that big library, which was about 2.5 million compounds. It was all plated. So in the assay, because we have to use radioactive alpha. So there was a lab was built especially for that. It is easy these days. You can have a corner room with perplexity glass, and you can have a radioligand lab, very easy, because it is very low radioactivity. Compound with 50 inhibition, I told you that we don't do every compound that curve which I showed you. It will be single point also. So 50% inhibition, if we saw the compound, we filtered out all the left. And computer also helps in a lot. Okay, then Ki was determined, and then 13 compound with Ki less than 1 micromolar we got, and then only 8 compound with less than 500 nanomolar. Now we are getting hit. You remember hit which I was talking last? So this is a hit now. So how many? 8 compounds, they were hit. Now from 8 compound we have to do lead optimization. I talked in the last lecture. So I'm just connecting to the last lecture. So what were the compound? So we had 8 compound. I'm showing you only 2 compounds. So we saw the compound. Now chemists are smart in that. They say, okay, this 2 molecule can be drug. 6 compound again, we filtered out. Okay, that we are not going to use those 6 compound. Just we went to 2 compounds. So we started working on left-hand side compound, which was, and I put this was pretty potent, 33 nano. It can be a drug actually. This compound can be a drug, and the other compound was 328 nanomolar. That was much weaker, almost tenfold. But I will tell you why we did not go with that. So the advantage was potency, disadvantage was selectivity in that case. If you go to other compound, we say advantage was selectivity, disadvantage was potency. So we voted to go for the less potent compound because it was selective. Getting selectivity is very difficult, but potency we can improve as a chemist. We published in Journal of Medicinal Chemistry the findings. So now we started optimizing. So we divided the whole molecule. Now little bit chemistry, so those who are not chemists, bear with me. So we divided the molecule in different parts. So you can see phenol, linker, piperazine, and benzyl. Every part we have taken and we have optimized, and then we all combine together to get a molecule. Next slides, 5-6 slides, very quickly you will see that. So first thing what we did, okay, right hand side, benzyl group. So we made about 80 compounds. No, actually, sorry. So yeah, benzyl group. First we took benzyl group. So we made about 80 compounds in that. Only halogen and 4 group of benzene was tolerated. You can see R is 4F, 4 chloro, 4 bromo. 4F, where it is 18 nanomolar. Okay, we got it. Okay, now we don't have to change anything there. So now let's move on. So only halogen at 4 position was potent. Fluorine in 4 position increased the potency, 16-fold. Removal of methylene reduce the potency. In between there is a methylene group, we removed that. Heterocyclic replacement with benzyl phenyl ring also reduced. So 80 compound, I'm not putting all compound, but these are the facts which was easy to understand for you, so I made that like fluoro at 4 position with benzyl group was fixed now. Now we solved one problem. Let's go to next. So now we go to other side. So this has 3-methoxy group and this is a benzyl group. So how we solve the problem here? So we made about 25 compound only. So we have several groups. So in this case, 4-fluoro was good. So we are going to combine all later. So you see that if you put chloro at 4 position, you gain some potency. Other substitution, they were not tolerated. So okay, I said okay, right side fixed, left side fixed. It was at that time, it was okay. Then we said, okay, now let's try parallel. So we started using, because there was some chemistries we can do very easily. So we said, okay, phenol, we can do some chemistry very quickly, where easy chemistry is there, alkylation is very well known. So we tried to do some library form. That did not give us very good, I will call it, result. But we got one compound which was 4-chloro. You remember, chloro was 288 nanomolar. But in this case, the quick chemistry we have done, and 4-chloro and 2CONH2 or a carbonyl amide group, that gave us a jump in the potency very quickly. So it became what, 5 nanomolar in that assay. Okay, so now we fixed right and left almost, but we made little bit more changes there. So 4-chloro and 2-amide carbon shows 7-fold increase in potency. Okay, now we have to fix the middle part also, this part. So we did some work there, but most of the work did not work out. The original which we had was the good for that. So although we have made about 60, 70 compounds, so it was not that good. So we just went back and we said, okay, we will keep OCH2CO there as it is, okay, which is, if you see, I mean is this. My cursor is not going there, so okay, this one. Yeah, okay, so this we kept it as it is. Now piperazine. Piperazine are well-known moiety in lot of drugs, and we have to really work hard on that to get it optimized. So we have left, right, middle. Now piperazine is left only. So the chemistry we developed, I will just show this chemistry part. I told you that I will just give you one chemistry slide. So this is how we made this molecule. We call it reactoamination, first reaction. Then you have to make it piperazine, so you have to use an amino acid, and then remove the bark, which is a protecting group, and then alkylate it through something else, and then you get the whole molecule. So this is the general route we developed. This route we developed was for medicinal chemist. When we made 200 kilogram, this route was changed by the other people who are called process chemists. Raise your hand. Yeah. Carbon, this is linker is COCH2. We have made longer carbon chain with more carbons also. They were not that active. So we checked everything possible. You are right, linker can be longer or shorter even. But we made, and this was the optimum. This molecule and this project could not take anything else other than this. So now SAR on piperazine. So we made several of piperazine. Those who know chemistry, this may be chiral also, R and S. We made all combinations, and it is much more difficult to do that. When you make chiral compound, it is much more difficult. What was pan out was 2R methyl group, and that was S was two methyl groups on the piperazine. One was S and one was R. But there was a problem with that. I will talk to you later about that. So we made about we made about 100 compounds, methyl at 2 position, increased activity, large group, benzyl group, and other things, other amino acid we used, but nothing worked out other than methyl group, because there was no pocket. When I showed you the computational model in last slide, in computational model there are pockets there, and you cannot fit everything there. So methyl was the only thing which can be fit there. So finally, 2R and 5S dimethylpiperazine increased the potency by 5-fold, which you can see here is number 3. Okay, this is 3 nanomolar. We made, like you asked me about linker, you can see that how many piperazine we replaced, different kind of piperazine, homopiperazine, piperidine, pyrrolidine, different type, and there are there, but none of them were as active as 2-methylpiperazine. So this was the optimum for us. So now if you remember, pay attention to this slide. We started with 328 nanomolar compound. We added 1 fluorine, so we got 3 nanomolar compound. We added 2 methyl group, we got 3 nanomolar compound. Then we added the chlorine on the left side ring. We got 1 nanomolar compound, which is very potent. 1 nanomolar is a dream of every drug discovery scientist, very potent compound. Then this was important because we added this, we call it urea. So this was reaching out to an amino acid in the receptor, and it was binding very tightly. So this gave us 0.8 nanomolar compound, which is like a picomolar compound, very, very potent. You need very small amount of compound. Okay, and then we removed 1 methyl because the solubility was a problem due to that methyl, because that methyl was on this side and it was masking this amine, this nitrogen, and we were trying to form a salt. So drug delivery now, you ask about, you have to form a salt for this kind of molecule. So because of the methyl, it was very difficult to form a salt because of the steric hindrance. So we removed that. Still we have from 0.8 to 1 nanomolar compound acceptable. Okay. So this compound was given a name. This became a development compound, which went into a preclinical and clinical trial. So this became our development candidate. I'll skip this slide. So we made it differently, as I told you, that we have to increase the yield, not 17%. So we worked with a process chemist, and he developed this whole thing, a whole procedure to increase the yield of the molecule, and which was like from 17% a medicinal chemist to 52%, and later it became about 78%. When we made in almost a 10 ton of compound, it became so they know I'm not a process chemist. Somebody else he was doing that, and I was working with him in Germany. We worked together. Okay, now pharmacological properties of this molecule. This is important, so everybody has to pay attention on that. So dog oral bioavailability. So this molecule was very nice, 4 milligram per kilogram. So it was orally available, 60% oral availability, which is, a dream of a drug discovery scientist. If orally given and still 60% compound is circulating in your blood, so it means that it is a very good compound. Half-life was 3 hours, not very long for this purpose. It was enough. We don't need very long half-life sometime. So 3 hours is a good half-life. Half-life means half of the compound is there in your body in 3 hours. Those were from pharmacology background. So T-half we call it, okay? Intracellular calcium, this is a functional assay. Functional assay we do to just find out that whether functionally it is antagonizing the receptor. One is inhibition, one is functionalizing. So this was very good, dose dependent. You can see from 1 nanomolar to 5 nanomolar to 10 to 100 molar, it was dose dependent. As you increase the dose, it is more active, this compound was. So, and we found the Ki, or Ki means inhibition concentration, is 5 nanomolar. T cell recruitment, because we are talking about inflammation, so whether T cell is recruited or not, this was also dose dependent. It worked very well in this model. So selectivity inhibits T cell recruitment by CCR1, that is what determined. Monocyte recruitment, so these are all the pharmacology we have done in this case, and they all worked very well. So selectivity inhibits monocyte recruitment by CCR1 ligand. So our ligand was extremely good to recruit that. So biological profile of BX471, so what we got so far? It was potent, 1 nanomolar compound in CC-ROD assay. Selectivity was 100-fold selectivity, again other 22 GPCRs. This is very important because there are other chemokines, similar chemokines, they are not hitting that. So this compound hit only the CCR1 and the selectivity fold, so we do it at another lab. In vivo oral bioavailability was still 60% and half-life was 3 hours. Looks like a good compound to put into animal now, right? So efficacy model, we did rat EAE model and rat heart transplant rejection model. I think I have the slide for EAE. EAE model is used for multiple sclerosis. And this was done at Stanford University. The compound which we put, cross reactivity in different species, as you see. In this case, human Ki was 1 nanomolar and rat was 120 nanomolar, which is good. I mean, in human it is more active. We don't care about rat. We are not developing a drug for rats, right? So high doses will be needed in rat efficacy model. That was a little bit of problem. But then we did whether this compound is safe for rat or not, because we are going to give a large amount of compound to rat. They can die. And we did that, so I think in rat EAE model, this was like 100 milligram per kilogram was very good. So we said, okay, we can go that. So we did the multiple sclerosis model, and you can see the parameters are there, 5 milligram, 20, 50, all rats survived. Treatment initiated after two days after induction. So the rat were induced with multiple sclerosis by a chemical means, okay? And this was dosed by subcutaneous injection and 3 times daily. Disease evaluated using a clinical score is set up by the pharmacologist. I'm not a pharmacologist, so they watch the animal, how they behave, and they put the scores. Okay, so clinical score, as I said, it was set up by the, so 0 is 1 tail is, limp in tail is 1. Similarly, different thing they watch and they see whether it is working or not. So rat EAE model of multiple sclerosis, you can see that it is efficacious in rat EAE model in dose-dependent manner. So 20 to 30, 50 milligram was excellent. So it reduces the scores, and the compound was safe to be used in animal at least. So heart transplant, I will just exit, go on that. So this was we published in immunology paper. This is not the topic today, it is there already. So it worked in heart transplant model also very well. So now breaking news is this, that this compound, it didn't work in endometriosis. I did not put the data for endometriosis because it was almost same, but there are 2 different diseases, multiple sclerosis and endometriosis are 2 different diseases. Both are inflammatory disease. After COVID, company was not very much interested, and I also left the company by then. And then this compound was in my radar all the time because it was very safe in human. When we did human trial, 300 milligram per kilogram, I think, or 1800 milligram daily dose was very nice in human. No side effect, which is very, very, very large amount of material. It was already on my radar, and I used to keep on talking people. Then COVID hit, and 2020, we found out that Germany, Berlin International Hospital, they started to treat COVID patient with this molecule. And Dr. Azhar knows that COVID is an inflammatory disease. And their thought was not to kill virus. Their thought was to reduce the inflammation, to use this molecule, and this molecule was very safe. So because virus has a life cycle, okay? So 5 days it is gone. But inflammation stays always, and it becomes worse if you don't treat it. So they used this molecule in 2022. This number is given at DRK. So this was a clinical trial was done by German government. They paid 3.2 million euro to Berlin Institute, and at that time this was repurposed. So this is a molecule which is really repurposed from another area to one area, and they did the clinical trial. The clinical trial was not very good. I will not say very good because it was the COVID was almost going down, so they could not get enough patient. So they did on 5 patients, 4 of them were treated, 1 was not treated for some reason, but they have to stop the clinical trial because they don't have the patient there at that time. Still, this compound is available with them, and if they want to do, if another time epidemic hit, they will use it. That's what they told us. So this was the breaking news at that time. This was the molecule, as you see, that they used the same molecule, BX471. So starting from Berlex in US, we did clinical trial for multiple sclerosis. That went very well, but we did not get the clinician. They tell that this is, they call it the score point. They did not get, there is some parameters they use, clinician, that this is very good. So number of patients were treated, but they were not enough to convince regulatory affairs that it is very good molecule. So like 20% patients were very good, 80% were not good. So that's why it did not go to market at that time. So this was surprisingly, I saw that. And at the same time, I was looking into this molecule all the time because it was very safe, efficacious, very potent molecule. And I was reading some literature. So I said, okay, we will look for more things. And then we found out that this compound is used in multiple myeloma in animal model. It was done by one of my colleague, actually. I'm not on that paper. And I talked to him. He said, yeah, I also left the company. Now I retired also. And he said, for 7 years, I'm traveling. I'm not doing any science. He's American, so American do that. After retirement, they just leave their home and travel all over the world. So I was in touch with Richard. He said, no, it was active. I did myself the animal model on multiple myeloma. If you want, you can go ahead and do whatever you want to do. I said, okay. So then a friend of mine who is in California, he came to Saudi Arabia. I showed him the data which I collected on the multiple myeloma animal model and everything else. He said that this is a very good idea to start a company. So he just registered a company in US called Renovo Biosciences, which is going to use some AI-driven drug repurposing also, and the normal drug discovery case also. And the molecule which he is going to use first is, you can see the same molecule, but he gave as Reno. It is not BX471 anymore. It is RNN first in class oral therapy. So somebody asked me why you call first in class. This is the first in class for multiple myeloma. And he has done extensive research on that, through an attorney, and he found that this is a viable target. And then he launched this company called Renovo. Renovo means renaissance. Renovo is a Latin word, so it is renaissance. And it is registered in the state of Delaware as a company. And these are the people who are involved in that. So Dr. Habib Hamidi, and Dr. Richard Kho are the founders, and I am the scientific advisor, and Dr. Ahmed Al-Askar, who is our hematologist, he will be the clinician. We are looking for one more clinician. Actually, they have hired some more people in this company, and hopefully this time we will be successful. That's our hope, inshallah. And they are really moving really fast. So now coming back to the last, talk I gave about academic and industry relationship. So these people are all from industry, but we have to go from industry to industry. They have to go to buyer and ask them to give us all asset, although it is dormant. कोई भी नहीं यूज़ कर रहा है उसको आजकल. कंपाउंड्स जो है वो शेल्फ पे लगे हुए हैं कंपनी के अंदर, तो उनसे कहा जा रहा है कि वो सब हमें दे दो और कुछ चार्ज कर लो या हमसे कुछ ऐसा एग्रीमेंट कर लो, 50-50 कर लो, 80-20 कर लो, लेकिन हमें सारा डाटा दे दो so that we can go and do a clinical trial directly in multiple myeloma. So Dr. Mana is also involved. He is doing NDA for us in Saudi FDA, and we will do, if everything goes all right, so we'll do first clinical trial in National Guard Hospital with under Dr. Askar's stewardship, and that's the plan right now for that. And many other people are involved. There are some attorneys are involved, but most of the work. This is all my data which I showed you exactly the way it is. I did not show only animal data because Habib asked me not to show that. Means multiple myeloma animal data, he said not to show that. You can see the confidentiality agreement is there. I have signed already a CDA, and both we and Dr. Askar, so I can show you only this much as a company founder. So in summary, we have developed a potent for small CCR1 molecule. This was, effective in animal models, but it did not do well in phase 2 clinical trial in at least 2 things. Hopefully, we are going to repurpose in multiple myeloma. These are the relevant papers. This is all on slide, so you can read that. This is many other people, including Dr. Askar, has written a review on multiple myeloma, and these are all my papers on CCR1 and patents also. I have about 10 patents only on this. So somebody was asking patenting, one molecule, 10 patent, why? Anybody? Not different purpose. Every carbon atom we have protected so that you cannot do anything on that. Every carbon atom, every hydrogen atom, we have protected in that. So that's why we have to file 10 patents. One patent, examiners, they are not ready to take because it will be maybe 6,000 pages. Every patent is about 200 pages. And the attorney comes in picture. So he was asking, so that's why we have 10, we can't have only one patent, a small molecule. But no, we have done such a nice work that nobody can enter into this field. And nobody has entered in this field so far. It is there for almost 20 years now. We are the only one who are talking to buyer to give us everything, nobody else. So that's how we have to do. And all this work is done in the Bay Area. I just wanted to show this picture, a beautiful place to go sometime visit. My company was right here in Richmond, and I used to live there. I still have a house there. So this is all about this work. Thank you so much. I think 40 minutes. Any questions? Yes, please. So very good question. Same CCR1 has 70 to 120 amino acid I showed you in one case. In human, it may be 120. In animal, it may be 70. But the active sites are usually same. Usually, why I say same? It may be there is difference between 1 or 2 amino acid here and there. So that's why usually it shows the difference between human and animal activity. So that's the reason. So the core of the receptor is same, but amino acids maybe they are different places also. So that's why it is showing difference in the activity.
Sir, what's the purpose of inhibition then?
Inhibition, you know how potent it is. Inhibitory consent, how you will know that it is potent. So you have to have some parameters for that, right? We step on the scale to see that what is our weight, why we do that. We want to know, right, that this is. So similarly, in case of enzyme, in case of receptor, in case of ion channel, this is the parameter that we call, some places it is IC50, some places it is KI, some places these days, I will talk tomorrow, I think tomorrow or day after tomorrow, about the new modalities where new things are coming, protein degradation. So PROTAC we call it. So I have a couple of slides I will show you in that. So that is called DC50. Degradation constant 50%. So protein is circulating in your body, you are degrading that protein, so how much? So you have to have some parameters for that. Okay?
Sir,
Yes, please.
sir, you said that it was also working for endometriosis.
Yes.
So why was this not taken up as drugs afterwards?
So in endometriosis also, it did not reach the good endpoint. Clinician determined that this has treated only 13% of the patient. Our company don't want to spend their money to treat only 13% of endometriosis patient. They want 100%. Reason, this is a big mystery in whole industry. Okay, that why some population is treated and other is not. Dr. Azhar is the right person to ask this question. He is a precision medicine person. So in case of precision medicine, you know subpopulation is treated and other population is not treated. So he will inshallah guide you on that.
I think this is all about personalized medicine, that and it is defined by a lot of factors. We all are different.
Yeah.
Some people say that it's because of the difference in the DNA. Some people they say it's different because of different ethnicities, which is due to different environmental, geographical features. So it could be multiple factors, but this is true that it's highly personalized. And if you can know what are those factors, yes, then you can address it.
We can address that. Like in case of Saudi patient population, they have a different genomics completely. They have some diseases they have only there. So this is called specific diseases. So we have to work for them only. This is a personalized medicine for genomics, and Dr. Azhar worked on that a lot.
So you said that you screened a million of compounds from
Yes.
your library.
Mhm.
How the screening was done? It was computational.
No, it is not computational. It is a real. We have robots, okay, and they do. So we have. I did not show that. So robots are there, and they will go pick up the plates. They will distribute everything, and the assay is set up in a way that it is very high throughput assay. And sometimes we do 10 compound in a well also. This is pooled compound, what we call it. So that's how it is done. So it is not done by human, it is done by robot. We have a $10 million facility in California. Most of them it is done by robot. But what we do, we don't pick up all 200 million compound. We sit down with these computational people and say that, okay, discard this one. And the robot, you have to direct robot go and pick up from there only, dispense into this plate, and then they will do the and usually actually initially we have done pooled compound, and after pooled compound we got the single compound. And single compound also I showed that 50% inhibition, so single point assay, not that curve which I have shown, which is 8-point assay, right? After single point assay, then we go to full IC50, which is 8-point assay. Dr. Zain will also talk about it.
Sir, if there is any drug that has gone through all trials
Mhm.
and it has came into the market and it is used by humans also,
Mhm.
then after some time we came to know that it is having ill effect on my body.
Mhm.
So that drug will be, what's the legal procedure that
That will be withdrawn. Just wait for my one lecture tomorrow. I will show you one drug example, what happened to that.
No, sir, what's the legal procedure for this?
Legal procedure is people will go to doctor, and doctor will tell to company that this is having this side effect and we cannot continue this drug anymore, and then it will go to regulatory or doctors or clinicians. They will send a note to FDA that it is not possible to continue with this drug because I have seen this thing in a patient or in population of patient. The regulatory authority immediately contact the pharma company who are making it, and pharma company will have a meeting, all those things with everything, and then it will be withdrawn if it is true.
Sir, then what's the need of
It is called
legal?
pharmacogenomics. The science of what you are asking.
Sir, I am asking that after going through all trials and then also it is showing ill effects, then
Uh-huh.
what's the need of
Not only that, it has gone through market for 7 years. One drug, Mylotrag. I will show that. And after 7 years, it was withdrawn from the market. Pfizer already made 25 billion because it went into patient. Okay, but it was withdrawn because in 7 years they found out that it has some side effect. Side effect you have to understand also. Side effect may be due to environment. Side effect may be due to some other medicine. Side effect maybe you are taking like आलू प्याज और दाल आप खा रहे हैं, उससे भी होता है side effect. So it has to be. It is a real science that how to find out how this side effect came. Okay. Any more questions, please? Okay. Yes.
This drug was did it help along with T cells and B cells, or was it working on its own? That I couldn't.
So this is a mechanism part of that. Okay. I will send you the paper where it works on T cell and B cells, and that is our marker actually, whether it works through the mechanism part or not. So you have to read that paper because it is a very heavy inflammation paper. It was published in Immunology Letters. You can understand more about that, or maybe tomorrow or sometime later I can explain to you. Okay. Any more questions, please? Okay, thank you. I think we finished 15 minutes over, and I took 40 minutes for this. I hope that you enjoyed it.
We shall see. This was a long session for you. So we are concluding for today's session, and for tomorrow, my humble request: please be on time, please start on time, and you will be getting the slides hopefully today. But don't worry, everything will be given at the end. And a little bit of telling you about the exam. Some of you might be thinking about exam. This is the requirements of the GIAN courses that you should be tested on what you learn. And it will be mostly multiple choice or fill in the blanks or true and false type of questions. No heavy writing stuff because we don't have that much time. And it will be all from within the slides that we have drawn today. Attendance would be sufficient, so don't be too anxious about it. And there will be a grade sheet that will be provided to you as well after the course finishes. We'll be sending it to you electronically. Right? So,
Yeah, just last
yes.
point I
Sir, the classes is supposed to have the Saturday starting with
नहीं वो
Oh, of course.
no, sir, because it is very hard work and lots of programs are there.
Just I want to say I forgot to say once yes the last Friday there is course marketing course basically there are no longer MBA as I almost last
But this was
that's okay but I'm just saying that the entire program
yes.
it is go
Don't
online that's
spread the
okay
word because he's going to be a multimillionaire very soon.
I'm not yet I wanted to we asked question that so we package the certain information with valuable points and intelligence we are going to package the name this compound not as a composition of metal present but as a use present for multiple myeloma it is already been filed
And buyer already have a patent for the same compound
for COVID. So this has 5 different areas.
Indications,
It
huh?
is patented. One molecule VX471. You type it
So
on
different
Google, and if you type on Google 471, you will find that about 17 companies are selling this compound as a tool compound. I didn't get any money for that. Okay, please.
I get one more message from Deksha that there is a lot of money in IP.
This is a lot of.
And it's a career.
It's a failure.
People are becoming patent attorneys.
Yeah.
So think about it.
But you have to learn the chemistry before you become patent attorney.
Yeah, you'll be very good at doing this work if you know the
Yes.
background. All right, so we conclude, and thank you very much for all your attention. People who are not here, that should not give you the wrong message. That mandatory the attendance is not mandatory. It is mandatory. It's your. If you're not here, it's your loss. Okay. All right. See you tomorrow then, inshallah.